Skeletal muscles, like the quadriceps femoris or the biceps brachii, have proprioceptors called muscle spindles to monitor the length of the skeletal muscle and body movements. Having this input allows the central nervous system to control muscle tone, posture, coordinated movements, and the corrective reflexes that help us keep our balance. When a muscle is stretched, the muscle spindles are stimulated and they transmit a nerve signal in a sensory neuron that travels to the spinal nerve, into the dorsal root and rootlets, and then into the spinal cord via the dorsal horn of gray matter. Inside the gray matter, the sensory neuron synapses with a motor neuron that innervates the quadriceps femoris. This chemical synapse results in EPSPs in the motor neuron until threshold is reached, causing a nerve signal. That nerve signal leaves the ventral horn of gray matter into the ventral rootlets and ventral root, back into the spinal nerve, on its way back to the quadriceps femoris. When it gets to the quadriceps femoris, it reaches a neuromuscular junction. There, acetylcholine is released from the presynaptic membrane, exciting the sarcolemma of the muscle and causing the muscle to contract. This contraction counteracts the initial stretching, helping to maintain the muscle's normal resting length. Notice that only one synapse is necessary in this integrating center. Therefore, we call this a monosynaptic reflex arc. If an interneuron is required, it would be polysynaptic. Also, since the effector is on the same side of the body as the stimulus, it's considered an ipsilateral reflex arc. Since it's easier for the quadriceps femoris to contract if its agonist, the hamstrings, relax, the sensory neuron also synapses with an inhibitory interneuron that causes IPSPs in the motor neurons that innervate the hamstrings. This action makes it harder for the hamstrings to contract and is therefore called reciprocal inhibition.